started with the specific objective of thinking through at early stages to the design of impact assessments on related to CGIAR innovations across a wide variety of innovations and parts of the CG portfolio. For some of you that were with us last time, you remember that so some of the presentations, like the one from Monday, will be on studies that are ongoing and are speaking to a particular method or a particular example. But then what we'll do today, and so we'll iterate over the next few months with the same logic, is kind of so every other month we'll give the floor to some early career researchers in the CG, with the idea of very much presenting very early ideas for impact assessment. So, half-cooked early work in progress, and with an open invitation for that to be, for anybody who identifies himself as an early career researcher, to take advantage of this opportunity too. So today we are very excited to have Kelvin from WorldFish and Tesfa from IITA kicking this off, and we hope to have other people with different approaches and different types of technologies kind of jumping in on this opportunity. So I guess the one other thing I should say is that today it happens to be the case that both Kelvin and Tesfa are going to be presenting some studies that where SPIA has been engaged in some of the scoping, but that's not what I wanted to emphasize is that going forward, that's certainly not meant to be the rule here, and so we're very much open to any ideas as a way of providing feedback, but also as a way collectively of starting to better understand what are people thinking about, how do we learn from each other, and how do we broaden the community of practice working on those things. So without further ado, and because we really want to hear from Kelvin and Tesfa, we're going to start with Kelvin. So we'll hear about a project that WorldFish has been working on. And so what we'll do is we'll have a presentation followed by a Q&A on that presentation, and then the second presentation on Aflasafe by Tesfa, and followed by the Q&A on that. And Ricardo will be rigorously keeping time to make sure that we have sufficient time to not just hear these initial thoughts, but also to collectively provide feedback. So with that said, Kelvin, the floor is yours.
Yeah, thank you very much, Karen, for the introduction and for the background. As Karen mentioned, my name is Kelvin Shikuku. I work with WorldFish, and more specifically with the Monitoring, Evaluation and Learning and Impacts Assessment unit. So I'm very honored to have the opportunity to present today, some research that we have conducted here at WorldFish, with the view to sort of create that foundation for potentially rigorous, evaluation of the genetically improved, farmed tilapia. So, I will be speaking about, what our approach is and what we've done so far, and also sort of a set of next steps. But before I do that, I also would like to acknowledge my colleagues whom we're working together in, in this particular project. Cristiano Rossignoli is the research lead for monitoring, evaluation and learning and impacts assessment at WorldFish. And Rodolfo Damlam is a postdoc also working with us. So let me now share my screen. So just as I mentioned a little bit as the slides upload, the outline basically starts with the
Yes, you can see now.
Okay, great. Okay, so just to give a little bit of an outline, so what you expect from this is that I will sort of speak about. I'll give a little bit of background, and then I will go into some objectives, or what are some of the challenges that that we experienced from previous attempts to evaluate impacts of GIFT, and then discuss a little bit what our approach is, and also present some of the results we have from a small study we've done so far, and then later on present our next steps. So a little bit into the background is that aquaculture is crucial for improved human nutrition and livelihoods. This is increasingly recognized. And the role of genetically improved, fish varieties in sort of boosting aquaculture productivity growth is also increasingly recognized, or well recognized. Now, tilapia. So among the many species of fish, tilapia is the second most important farmed fish globally. It is estimated that about 5.5 or more than 5.5 million metric tons of farmed tilapia produced every year. And the genetically improved farmed tilapia variety, which I will be focusing on in this particular presentation, was developed by Wildfish, formerly known as ICLAM, or the International Center for Living Aquatic Resource Management, and its partners. Right, so that work started sometime back in the 1980s, and also in the 90s. So the GIFT strain is faster growing. It has high yielding ability, and also with a high survival rate. And also it basically tolerates a wider range of environments. So those are sort of some of the desirable attributes of this particular variety. Now, in Bangladesh, fish contributes more than 60% of the dietary animal protein, and the country, Bangladesh, is the fourth largest tilapia producer in the world. And so and that also explains a little bit why we choose this particular context, but other reasons I will explain also later. So the genetically improved farm tilapia was first introduced in Bangladesh in 1994. After that, there has been several improvements, right, on the strain, and therefore further dissemination happening in 2005 and also 2012. From our records and from other studies we've done, we observed that most hatcheries were established, during the period of 2011 and 2015. But we also, there are many other hatcheries that were established before this particular time period. So the contribution of GIFT, not just in Bangladesh, but worldwide, is widely acknowledged. But in Bangladesh, we particularly see that the GIFT technology is the tremendous improvement or growth in tilapia industry in Bangladesh is to a very large extent attributed to GIFT. So, I mean, this particular figure you see here shows, for example, in 2010, production was less than 25,000 metric tons, and you can see that has grown quite a lot. By 2018, it was more than 300,000 metric tons. Given this potential of the genetically improved farm tilapia, given that the fish has been introduced for a long time, so there's sort of some sufficient diffusion in this particular context. So what have previous studies tried to do, to understand impacts of GIFT? So previous work, and mostly based on on-farm, on-station or on-farm trials, have suggested that growing of GIFT increases yields and therefore, sort of boosts incomes. It reduces production cost and also has the potential to generate rural employment. Now, these studies shed some light, and I think to some extent are very insightful in sort of informing us what are some of the mechanisms through which GIFT could potentially generate impacts. And so they are very useful to that front. But then they also suffer some data and methodological limitations. One, because as I mentioned, they are sort of based on on-station and on-farm trials, and therefore may not necessarily reflect, the farming context, right? And farmer management, growing of gift under farmer management practices. They also tend to suffer from issues of power, so they are often underpowered, and so there's sort of a limit as to what, how much of impacts evaluation can be done using those data. But also there's then a bigger issue here, which is sort of related to misclassification of strains. With farmers basically, from what we understand, is that they basically don't know what varieties they have in their farms. And so without sort of devising approaches to sort of try and make sure that we avoid that misclassification problem, we may end up in sort of measurement error issues. And so there's therefore a need for a rigorous evaluation of the causal impacts of gift. And at the moment, therefore, there's a big gap, in terms of understanding really what the impacts of gift are. And here we are referring to the longer term impacts. So now turning to this particular study, the current study, we have some information, at WorldFish. We know, for example, that the dissemination of gift varies spatially and also over time. We have records, for example, tracking the dissemination of the genetically improved farm tilapia. If again I can discuss a little bit here what the seed system looked like or how gift was introduced in this particular context. So you can think of, you have at very high level some you can call them like broodstock multiplication centers. Those are hatcheries that receive elite germplasm from, let's say, WorldFish, and they're supposed to multiply the broodstock for subsequent dissemination to other hatcheries in the private sector, who then multiply it for dissemination to grow-out farmers, okay? And so we have records about when exactly those farmers received elite broodstock and what strains they have. And so there's some variation here in the data, even among the strains that are cultured by hatcheries in Bangladesh, in the sense that even for gift, we have some hatcheries that are sort of producing and disseminating sort of the latest generation that was disseminated in the country. But there are also hatcheries that are sort of continuing to disseminate the older strain of gift. So you can then see that there's variation here, not just spatially and over time, but also in terms of the even within the gift strain itself, there is variation in terms of old and new. So like I said, this information is useful in a sense to help us sort of track dissemination and also to identify strains. But the question really is, can this information be mapped to farmers, right? Because we want to collect data at farm level, farm household level, to be able to evaluate impacts. But can we really map this information from hatchery level to farmer level? And so that's the question sort of we try to address in this particular study. And so in other words, you can think of, we are trying to define here, what are the catchment areas for gift, okay? And catchment areas, of course, we look at location of hatcheries. We are concerned about the volume of seed that is sold. We are concerned about the number and geographical location of tilapia farms. So we had discussions with SPEAR, for a considerable amount of time, trying to see how best we could sort of design a rigorous study, to evaluate impacts of gift, and which then led us to sort of submit a proposal for SPEAR Track 2, which is a smaller grant to sort of map out the catchment areas. And validate those catchment areas. But basically the question was to see whether it was really possible for us to map the information of the hatcheries back to to farmers. So the objectives then of that particular study were to describe and document the dissemination process for gift via hatcheries to farmers, and the implications for how hatchery catchment areas could be defined, both conceptually but also empirically in the context of an impact assessment study. And then, like I've mentioned, was also the second objective to validate, the catchment areas as defined by hatcheries, so we validate that by collecting data directly from farmers. Now, to answer these two objectives, we took a two-step approach. So in the first step, which is basically to identify the catchment areas, we list all the tilapia hatcheries, both gift and non-gift. So it's like a complete list, so a census of tilapia hatcheries. We work with a partner in Bangladesh to validate that list and see who is active, or who basically collect contacts, for example, so that we could reach them for the interviews. And after doing that, then the next step was for us to contact those hatcheries for interviews using the computer-assisted telephone interviews. So here is important to mention that although we list all the tilapia hatcheries, and there are about 200 or so, we are able to reach 134 hatcheries for interviews. And in addition to that, we also have key informant interviews with experts in Bangladesh who are knowledgeable about tilapia dissemination. Now, step two was conducted only after step one was complete, because we first needed to understand where are the catchment areas, okay? And then within those catchment areas, we list all tilapia farmers. We generate lists of tilapia farmers within the catchment areas. We draw then a random sample of 3,000 farmers for interviews, okay? And then we are able to reach about 2,956 farmers, okay? But this second step was done only after knowing what the catchment areas are. And so just also to give you a sense of what type of data we collected, at the hatchery level, we look at, we sort of rely on recall data and ask them to tell us how much, where they've been selling seed over the last 10 years, right? So starting just before 2012 and then coming up to 2020, for each year we ask them about the buyers of the seed. We ask them about the districts and the upazilas where they sell the seed. So upazila here is sort of, you can think about the administrative structure in Bangladesh. You have division at the high level, you have districts, and then there's union and upazilas, and then before you reach the village. And so it's a level closer to the farmer, but there's still sort of another level in between, which is the village. But we couldn't track dissemination to the village level, so upazila was the lowest level we could go, but it's still very close to the farmers. So we ask about those hatcheries. Although Wildfish has maintained some database, we still ask them about the sources of the breeder seed. So where they obtain their breeder seed, have they replaced, the breeder seed? When did they replace, and where did they get the replacement from? So such questions. We also ask about the quantity sold, and we ask about the type of seed that was sold. And here we don't just have like two broad categorization of gift and non-gift. This group of non-gift comprises several other strains. Okay, and then at the farmer level, we then collect data about the location of the farmers. And so for that one, we go up to the village level, but for purposes of matching and defining validating the catchment areas, we use data at the upazila level. And then we collect again, we ask them to tell us the name of the source of the seed, okay? So for example, not just saying they bought from a hatchery, but also saying whether they know the name of that particular hatchery. And then, unlike the hatchery here, for farmers, we focus on the last 5 years. So we try to ask them to recall how much seed they bought during the last 5 years from the respective sources they mentioned. And then, in addition to that data, we collect information about demographic characteristics. And so, of course, as you can imagine, there's recall recall bias here. But at the hatchery level, we worry less because that information could be validated also by some information that that WorldFish has been collecting over time. Now, so turning to the analysis then, so we perform sort of 3 levels of matching. So again, if you recall, our objective is to define the catchment areas and to validate those. We perform 3 levels of matching. In the first level, we basically ask, you know, look at a farmer in our sample and ask, so define a geography. So let's put it this way: define a geographical area where seed was disseminated, right, by the hatcheries in our sample. Okay, and then we ask the farmer whether they, I mean, we look at whether the farmer is mentioning that they obtain seed from a source that is within that particular region. Okay, so it's a very high level of matching, as you can imagine, right? So we would almost expect that all of our hatcheries or all of our farmers would be would fit within that particular set. But then in the second level, we then ask, okay, we have farmers that we survey, and so we check whether I mean, we have hatcheries that we survey, and we ask the question whether farmers are mentioning at least one of the hatcheries that we have surveyed. Okay, so in this particular case, our interest is to see whether or what proportion of the farmers are able to at least mention a source of seed that sort of corresponds to the names of hatcheries we have surveyed. So as you can imagine, the number of hatcheries then reduced because we have a tighter restriction there, and I will show those results in shortly. But at the third level is stricter, is even more strict in the sense that now we ask, okay, so consider a farmer who is located in Upazila X, and recall again, I mentioned Upazila is sort of a level that is very close to village. So a farmer is located in Upazila X, they mention a hatchery as a source of, so they give us the name of the hatchery as a source of seed. And then when we ask the hatchery also confirms that they sold seed in that particular Upazila. So it's sort of farmers who are mentioning that they obtain seed from a particular hatchery. If we checked with the hatchery and the hatchery says they did not sell in that particular Upazila, then they would not, for example, fit in this level 3. So that's why I said it's a bit, it's more strict compared to the other 3 levels. I mean, the other 2 levels, but it's a level where then you begin to have a tighter matching, at least from how we see it. So just to then give you a sense of how, from a graphical perspective, how the diffusion of gift looks like between 2012 and 2020. So what you can see here is that in 2012, for example, there are a few hatcheries, and you can also see that in terms of the volumes or the quantities of seed that is disseminated is not that much. But you can see that as we move towards 2019, we don't just have more hatcheries, but also the distribution is sort of they are widely distributed across the country, and also you can see that the quantities that are being disseminated are also have also increased substantially. So this also just paints the picture that the diffusion of gift has indeed happened over time, and we have sort of good spread in Bangladesh. Okay. Now, as we begin now to look at whether we can define catchment areas, this particular bar chart shows basically places where we have most of the dissemination of gift happening. But even more important is you can see that this particular chart corresponds to the maps I just presented before, because you can see that most of the dissemination has increased in recent years. And so you can see, for example, when you look at 2018, 19, and 20, that those are sort of the bars that are higher relative to 2016. And so here we look at only the last 5 years, although we have data, as I mentioned, up to 2012. Okay. But this also again just reinforces the variation that we see in the data, especially, but also across time. So again, going into the catchment areas, as I mentioned, then we are interested in what's happening at the unit that is very close to the farmers, and in this case, the Upazila. So here I present, for example, on the left-hand side, you can see that these are the Upazilas, where the hatcheries in our sample mentioned that they've disseminated gift tilapia, okay? So both gift and non-gift. So these are the places where tilapia seed is reaching. And on the right-hand side, you can sort of think about this as where the farmers that were interviewed in our sample are located. Okay, so it's because we don't do a census of the farmers. This is basically showing the geographical location of the farmers that were interviewed in our study. But then, as I said, now let's look at the results of the matching. So at level 1, as you can see, we have more hatcheries. We have more farmers again, 1,900, and we have more Upazilas at this level, which is 80. If you remember again, here is level 1. We are just saying, okay, a farmer is mentioning at least that they obtain seed from a geographical area where the hatchery is disseminated seed. But what is interesting then, as we move to level 2, as we would have expected, is that the number reduces a bit, for gift as well as non-gift, but we still have quite a high number there of farmers, as well as the number of Upazilas almost reduces to half as we move from level 1 to level 2, both for gift and non-gift. But as you move to level 3, then you clearly see that it sort of begins to give us a more accurate match. But then the number of hatcheries reduce again, and the number of farmers shrink, and again actually the number of Upazilas reduces even more. What we also observe is that at this particular level 3, then we also don't have non-gift hatcheries or non-gift farmers or Upazilas at all. Okay, and again, as you can observe here, we have fewer non-gift hatcheries than gift hatcheries, and that reflects the situation actually as it is, because mostly the non-gift dissemination model is dominated by large multinational companies, which are not many, but they have sort of a wide network of agents who aid in the dissemination of the seed. Okay, so geographically they're still able to reach a good distribution, but they are not as many in numbers as the gift hatcheries. So just again to give you sort of from a graphical perspective how the matching at level 1 looks like. So this map basically shows again the places where the geographical areas where the seed is going, and farmers are mentioning that actually they bought seed from those particular areas. So that's what you see in the green on the left-hand side for gift. And on the right-hand side, you can also see, what's happening in terms of the non-gift. So you could say, like at this level, we still have a good number, both in terms of farmers and hatcheries for gift and non-gift. But it's a very rough level, and still difficult then to define, I think, a counterfactual at that level. Now at level 2, then we begin to check where. How many or what proportion of farmers are mentioning at least one of the hatcheries we surveyed as a source of seed. And as you can see in the results presented here, is that most of the farmers, mention, are able to identify a source that is among the hatcheries that we interviewed. Okay. But this particular graph also shows you that apart from the hatcheries that we survey, there are other players also in the seed system that play a critical role in the dissemination of tilapia seed. And so, for example, the commission agents are used mostly, as I mentioned, by the non-gift hatcheries. But also recall, like I said, we were not able to interview all the hatcheries, so we also have this group of farmers who identify other hatcheries that are outside our sample as being the source of seed. Okay, NGOs and nursery, not much in terms of being a source of seed. So how that looks like, again, from a graphical perspective, you can see it here again for gift and non-gift. Again, the green shows the places where we are able, on the left-hand side, where we are able to identify a match for gift tilapia. So a hatchery, a farmer is identifying at least a hatchery within our, that was surveyed in our study. And on the right-hand side, you can see what the story is in terms of non-gift. So we still have a good number of hatcheries. I mean, there are few, but you can see that the distribution is not sort of concentrated in a particular area. It seems to be a bit distributed, except for the central region. Okay. So now at level 3 is, again, as you can recall, is where now we look at sort of exact matching, right? The farmer mentioned the hatchery, and the hatchery confirms that they actually sold seed in that particular area where the farmer is located. So at this level, again, as the table showed, we still have a good number of gift hatcheries and farmers. But then we begin to sort of lose the non-gift, the non-gift farmers and hatcheries. Okay, but this is the level that we would sort of, desire to sort of be able to construct our counterfactual there. So then, just then to conclude and sort of also give what our next steps are, in this particular study that we've conducted, we are able to sort of have an enhanced understanding of the diffusion, and we see that there's a sufficient diffusion of gift indicating suitability for an evaluation of long-term impacts, as because gift has been disseminated for a longer time in the country, and also the diffusion seems to be quite spread. And then we have also been able to identify and validate the gift catchment areas, as of course, as we've seen for non-gift at that third level. We don't see many players, although that's the level we desire to sort of construct our counterfactual. And so we sort of, in terms of the next steps, we are thinking about this in terms of what would be our immediate next steps and what would sort of be then sort of longer term steps. So as the sort of immediate next steps, we are thinking about trying to sort of follow up some hatcheries that we missed in the survey, with the hope to see whether there's new information we could get that will help us to sort of utilize the information at the third level of matching. Okay, but then we then want to use this particular information that we have gathered in the study to be able to construct a counterfactual, especially because we are now able to sort of match, map information that is, provided at the hatchery level to the, to farmers. And then another step, next step is to identify the estimation procedure. So we are thinking of matching because the hatcheries that were identified, for example, as the entry points for the dissemination of gift in Bangladesh, there was a clear criteria defined for those particular hatcheries, and it's very well documented. So, we know those observable characteristics, what they are. But we're also very open to suggestions about other potential approaches we could use in, as we develop the proposal. And then in terms of outcomes, we are thinking about income, food and nutrition security, poverty, and also we would like to think about some proxies from an environment front to understand, whether there are some ecological trade-offs as well associated with gift adoption. So here I highlight just a few in terms of less water and input use, but we are also exploring other potential environmental outcomes. And then we observe a lot of heterogeneity in our sample, not just by farming systems, but also by important socioeconomic characteristics. And so we are would like, as we move into the next steps, also be able to design a study that will help us to understand those distributional impacts of gift. So so I'll stop there and welcome any comments or suggestions from the audience. Thank you very much.
Thank you, Kelvin. I mean, this was a very comprehensive presentation, and now I think the floor is open, and especially for providing input and suggestion, how can Kelvin improve these initial ideas? So the floor is open. Please raise your hand if you want to. Yes, Srini.
Hi, Kelvin. How are you? It's Srini from Nairobi.
Hi, Srini.
Yeah, a quick observation. I mean, regarding your outcome. It looks really many, I guess. You may like to just to have your primary outcomes to make it much more clear what exactly you wanted to see your primary outcomes. You know, because most of the outcomes are much bigger and broader because I don't know, your sampling analysis is going to be based on this. So you need to be clear on your primary outcome. The second thing is matching. I mean, I'm still not clear why you opted for this matching method. Probably just a little bit. I know you talked about, but still it is not really clear to me. Yeah. Thank you.
Okay. Yeah.
Thanks, maybe we can take some 2 or 3, Kelvin, and then you can react to that.
Yes. Yes. Perfect.
Okay. Comments or suggestions for Kelvin? Maybe for now, while people are thinking in their questions, maybe Kevin, you can react to Sirini's comment.
Yeah. Okay. Yeah. Thanks, Ricardo. Yeah. Thanks, Srini. I think I mean those are excellent questions, and also thanks for the excellent suggestion. So I think your first point on the primary outcomes, I think that's well noted. So why we mention these particular outcomes is, first of all, looking at, I mean, for example, some of the studies I highlighted as having already been done to try and assess the performance of gift, sort of have sort of set stage in terms of what we can see as the causal pathways, right, for gift. And so, for example, increased productivity translating to incomes, for example, or, consumption of diversified diets, for example, contributing to nutrition security. So I mean, in thinking about these outcomes, we were sort of coming from that perspective. But you're right in the sense that we haven't sort of decided on what the outcomes look like. So we may have to sort of separate this in terms of what are the primary outcomes and what are the sort of secondary outcomes. So that's very well noted. Thanks for that. I think in terms of matching, I think for us, we were thinking first of all in terms of, like I mentioned, we sort of have a clear criteria sort of defining, that was used to select some hatcheries that were targeted for the initial dissemination of gift, for example, in Bangladesh. And that criteria is uniform across all the entry points for gift in Bangladesh. Yeah, so that was sort of giving us also some confidence here, is that we are not just matching for purposes of matching, but it's sort of we are starting from the point of view that we have some observables that were, clearly defined from, from the beginning. Yeah, but we, like I mentioned, we are open to other procedures. I think as we move into the next, next steps.
Yeah. The hand of Carlo.
Carlo, Ricardo, can you just?
Sirini, could we give time to others to also participate? We're running.
Yeah.
Carlo, go ahead.
Yes, thanks. Carlo Zari from IFPI. Just a question actually for the next steps. So you mentioned that you're going to use this information for matching based on observables, but in the future, how you will control for possible sample selection bias, given the fact that farmers who choose to have a tilapia farm, an hatchery, will be, let's say, selected based on systematic characteristics that make them different from those who don't adopt tilapia farming. So how can you select? How can you essentially match for based on unobservables? Thank you.
Yeah. Yeah, I think is an excellent point. I mean, and I think clearly one that we would have to face, right? So, I mean, I can see it sort of two ways. One is, there are some characteristics you can imagine that sort of diffusion of gift, will also, will sort of also change those, some of those characteristics. And as you know, then in that particular case, then, those would be sort of not suitable for inclusion in the matching, and of course it's difficult to rule out. So I guess what we could do here is to, try to really check whether there are some variables that we could say, well, those characteristics don't seem to be, they seem to not to be likely to change, but because of, let's say, introduction of or diffusion of gifts. So I think that we are not sort of looking forward to just sort of throwing in any variables, as you correctly have mentioned. But I think the other thing we could do here, and that's why I was speaking about potential for, let's say, combining not just matching but also other other procedures. So we were thinking of not just relying on one particular estimation approach. As you correctly mentioned, there are limitations there even for the matching, but we were thinking in terms of also using some other estimation procedures, either in terms of robustness analysis, etc. At the moment, we are still in the process of trying to, work with, with SPIA and, and others to see how, what other approaches we could use. Yeah, but we are very open to suggestions.
Karen.
I don't know if Cristiano, you may want to add something, because Cristiano is also in the meeting. Cristiano, would you like maybe to?
Kevin, can we go listen first to Karen, because she has raised her
hand. Okay. Yeah.
And then Cristiano can interview.
Yeah, just thanks, Kelvin. And I think I'd like to make a broader point here, because I think the discussion we're having is very relevant for many different innovations in the CGIAR. So it's important to make a distinction here between the two different levels at which there is data. So there is data at a hatchery level, for which we know, for which World Fish has the criteria for why those hatcheries received or not a gift. Okay, and so there is hatcheries identified that didn't satisfy those criteria. Then the impact, of course, we're interested in is not the impact at the hatchery level, it's the impact at the farmer level. And the whole point of the exercise that Kelvin presented was to define, to see whether it is possible to link farmers to hatcheries, so that the variation that, the whole idea of identifying strategies is to think about, okay, what are the sources of variations and which ones of those sources of variations are exogenous. So World Fish has information of a source of variation, both whether the hatchery was selected, but more importantly, when those hatcheries got different types of gifts. Okay, the exercise that was done in the scoping was to see can farmers be linked to hatcheries, and then clearly and this is the point I want to make, because that's a more general point. The question will not, we know, and we shouldn't aim for trying to figure out which farmers select into getting information, getting data from, getting brood stock from certain hatcheries or not. That is endogenous and driven by observables, and no matching is going to solve that problem. But if it is possible to define, if the geographical nature of catchment areas is such that we can predict with reasonable accuracy which farmers source from which hatcheries, then the variation at the hatchery level can be used as a source of variation also at the farmer level. And so that's kind of the logic of the exercise here, to be refined and to see then how this can be linked with outcomes that change over time, etc., which is very much the next step. But so I think that is the part also that is relevant for many other kind of diffusion of CG innovations.
Any reaction, Kelvin or Cristiano, or can we ask for another final question?
No, I think Karen clarified perfectly this point, so no need for me to add anything here. Thanks, Ricardo.
Ricardo, there is a question in the chat from George.
Okay, maybe James first, and while reading this question.
Hi, Kelvin, thanks. Just, I guess my question is about the timing, and whether the current defined catchment areas, Karen just described the logic of doing this, defining these catchment areas, and I just wondered whether you guys can, I might have missed this, but like whether earlier on in the process, the more entrepreneurial farmers were willing to go outside of their catchment areas that are currently defined. So say earlier on in the process was when there was a kind of early introduction where the farmers traveled further, and I don't know whether that complicates things for those time periods, or in terms of some of the outcomes that you're interested in, because there could have been what currently represents a catchment area may not have been kind of de facto the geographical basis from where people may have been willing to travel further earlier in the rollout, if you sort of mean. So I just wondered whether that's something you guys have thought about and looked at.
Thanks. Maybe let me start, and maybe Cristiano can also weigh in. We thought about it because, for example, when I was mentioning that we observe some differences, especially in terms of the different farmers that are growing the different types of strains. So we observe, for example, already that the farmers that are growing the non-gift strains seem to have, for example, a relatively higher wealthy index, and they have, sort of larger farm sizes, etc. If those can sort of give a little bit of a proxy about the sort of the wealth status, then we could sort of begin to see. Thinking about the level 3 again, because this I think also becomes relevant for level 3. We were thinking that perhaps one of the reasons we haven't seen, non-gift farmers at that level is also possible because of the reason you mentioned. Is are they, for example, sourcing outside, those particular catchment areas? For example, they can afford to source from a distance, or they have some arrangements for delivery of seed from further distances, and so we are not able to catch them within those defined areas. So yeah, I think that's a possibility, and we see that it's likely to happen more in the non-gift with the non-gift model than the gift model.
Kelvin, I have a final question here from George at Seaford. How are you taking care of recall bias, especially when record keeping isn't good?
Yeah, Yeah, it's a good one. I think at the hatchery level, as Karen mentioned and also as explained in the study, we sort of started with some because we also track in some way we also track sort of the dissemination of seed from from different hatcheries. So we have sort of a database that can help us to track that dissemination to some extent. But I agree. I mean, especially in this particular study where we work sort of 10 years back, and so there is, as I acknowledge in the study, in the presentation, there is clearly some recall bias, I would say. But the fact that sort of that information that they are reporting could also be validated by records that we also keep, at Wellfish, can help to some extent address this issue. At the farmer level, I think is a different story because we don't have, we don't have such records. But at least at the hatcheries, to a large extent, we are able to cross-check that information. Yeah, so we haven't done it yet. The information that I presented here is not cross-checked against, for example, what we have in our records, but that's possible to do at the hatchery level.
Thanks, Kevin. I think for giving time to Tesfa, I would like to stop here, and I would like to thank you for your presentation, and also encourage the people who have other input and suggestion for Kevin, please communicate directly with him and bring those suggestions forward. The idea is to start this conversation to end up with a very good proposal in the future. Thank you very much, Kelvin and team. And I would like to invite Tesfa then to do his presentation.
Okay, thanks a lot, Ricardo, and thanks everyone for the useful comments and suggestions.
Go ahead, Tesfa.
Hello? Hello,
Yes.
Can you hear me now?
Yes.
Okay.
Presentation.
Thank you, Ricardo, for inviting me, and I'm also, like Kelvin, very happy to share some of our ideas on impact evaluation of Aflasafe, because we are currently at early stage in the process of this impact evaluation. Actually, we are starting the process of data collection. I framed it as institutionalizing, tracking, and monitoring of the dissemination of Aflasafe in Africa. But my presentation will actually focus on why we are collecting this data and what's relevance for impact evaluation or our planned impact assessment, and how are we planning to use this data for future impact assessment. So let me introduce myself. My name is Tesfamicheal Wossen. I am an economist with IITA. I'm based in Nairobi, Kenya. And I'm presenting this impact assessment work on behalf of IIT's Aflasafe team and socioeconomics team, and many others, including SPIA, who were involved heavily in the process of this proposal development. Seems it's not moving. Okay, it's going now. Okay, so I will give you a brief background about aflatoxin and also our journey from the initial idea of developing a proposal for evaluating Aflasafe in Nigeria to the current status, where we are actually discussing for a small grant for data collection, as well as some of our ideas on impact assessment and the next plans, and also some few remarks and some pointers for feedback from colleagues. Aflatoxin is, in general, an invisible poison caused by fungus. I think it's called Aspergillus flavus, and consumption of contaminated aflatoxin-contaminated food actually causes in some cases death, but most cases health issues, especially liver cancer, stunting, and so on and so forth. Not only health effects, but also it causes it has an impact on income because it affects not the productivity per se, but the quality of the production, and also it actually affects the overall economy, especially for tradable goods, because of the high level of sanitary regulations in high value markets, especially for export commodities like maybe not necessarily maize in some cases, but for some countries, maize is also a tradable commodity. So this Aflasafe is basically a biocontrol using the same strain of fungus that actually outcompetes the poisonous fungus. And in doing so, actually reduces aflatoxin contamination by about 80 to 100%. This is the evidence we have from ongoing efforts and past evidence efficacy studies by IIT and other colleagues. I must say that Aflasafe is a bit a unique innovation in a way, because unlike other CGIAR innovations, uptake is actually driven by mostly health or nutrition benefits instead of the profit or productivity in a way, particularly for non-tradables, but like meth. But for tradable commodities, some profit actually could be a driver, especially for export commodities. The other issue is the credence nature of quality, or the unobservability of aflatoxin. This which requires actually some regulatory interventions in market development and incentive structures to drive adoption. And the other unique characteristics of Aflasafe, like basically biofortified crops, is also sometimes benefits may not be instant in a way, because the effects occur over time. Particularly the health effects and the nutrition effects, but then the negative health effects may actually persist over the lifetime of individuals, and this actually poses a challenge for evaluating impacts in terms of articulating and quantifying benefits and costs. And also an additional property is that dissemination or commercialization mostly relies on the private sector or the market structure, not really the public sector, like improved seed varieties, which are a dominant innovation at the CGIAR. So to give you a brief outline, I actually started this idea, the idea of evaluating the long-term impact of Aflasafe in Nigeria in 2017, around 2017 and 18, when I was a postdoc in Abuja. The idea came with my interaction with the agri-result monitoring and evaluation team. There was this agri-results is a project that scales Aflasafe in Nigeria through the pay for performance mechanism using the aggregator model to farmers, especially to susceptible areas, mainly for maize, and I think Nigeria is mainly for maize. So our idea was actually, since there was some kind of dissemination in Nigeria, actually through this agri-results project, we were thinking of kind of piggyback a bit on the ongoing project and evaluate the impact of Aflasafe on farm level or household level outcomes, focusing on income, behavioral change, and also downside risk related outcomes. But then then we developed this proposal in response to the SPIA's call for expression of interest for long-term impacts for CGIAR innovations. As you might expect, then the SPIA came back to us and with a suggestion that we like the proposal, we like the innovation. We think that it's it is an innovation that needs to be that requires evaluation. But then at the moment, then we are not sure really if there is an evidence on large scale uptake or dissemination actually to evaluate in Nigeria. So we couldn't actually provide evidence on the extent of the dissemination or the extent of uptake to actually conduct a reasonable ex post evaluation of the impacts at the farm or household level. So we had a follow-up discussion with SPIA, particularly with Karen in Rome, and the idea was, okay, then if we could not actually conduct an ex post evaluation, maybe we could look into a forward-looking kind of evaluation focusing on scaling strategies, because the innovation is also relatively new compared to other CGIAR technologies, particularly for countries like Senegal or Tanzania, not so much for Nigeria. So we plan to actually then incorporate more of a forward-looking type of evaluation design, focusing on commercialization strategies, particularly on markets related and demand creation related incentive structures on top of what the agri results was kind of like implementing back then in 2019. So we were kind of targeting that matchmaking call, and also like SPIA learning oriented impact study call. But then we still have an issue, a recurring issue, because even for designing commercialization strategies, we need to actually understand previous rolling out mechanisms and understand what the effects of those rolling mechanisms were, and then base our interventions or scaling strategies that we propose based on the lessons from previous efforts, which we couldn't do at the time, because the agri results project actually used more of like a similar rolling out mechanism for using the aggregators model. And we couldn't actually get some more relevant information or to actually compare the cost effectiveness of the alternative scaling strategies. So the idea then was to actually get more and better data on previous dissemination efforts and also rolling out mechanisms. So we then continued our discussion with SPIA, IATA, and the AF4NH, including the IAT's Aflasafe team, the SPIA team, and also the socioeconomics team at IATA. And the idea was then to actually use a small grant actually to get better data on past dissemination efforts, rolling out mechanisms, and the status of production and dissemination across not only across countries, but also within different locations within a country. So we started this discussion with SPIA in late December with the idea of developing a routine mechanism for tracking and monitoring of past current and also future Aflasafe dissemination efforts in target countries, mostly in sub-Saharan Africa, Western and Southeastern Africa. So the idea is to use the data not only for ex-post evaluation, ex-post impact assessment, but also to lay the basis for designing future and more rigorous accountability-oriented impact studies using this data as a basis. So we had subsequent discussions with SPIA, especially on the type of data, what kind of indicator we should collect, we should target, and why should we collect those indicators? What is the relevance for our impact assessment? And if we want to collect those indicators, what kind of outcomes are we thinking actually for evaluation? And defining all those indicators. So we came up with a kind of classification of countries basically into 4 groups based on the development and commercialization of Aflasafe. So we categorize countries into countries where full commercialization is already undertaking, like Nigeria, for example, where there is production capacity and manufacturing and distribution channels already existing. Also, like some few countries where the development of Aflasafe is ongoing at the regulatory approval stage or licensing stage, and some countries where the Aflasafe team is at the piloting or testing stage. And we also had another category where no testing is currently being undertaken, but there is some kind of Aflasafe importing for use in the domestic economy, and the idea being then at the end, then we'll have a kind of database where we could actually track the production and distribution of Aflasafe within and between countries over time through this system. So probably we'll have some data like this map, not as fancy as this, but probably something like this. So then we came up with a discussion with SPIA and other colleagues. Then we came up with basically 3 important indicators to consider in this effort. The first is to collect important indicators to actually document the spatial and temporal dimension of Aflasafe dissemination across the different locations in Africa, or mostly in target countries, not the whole Africa actually. So the plan is then to for each country then to look into whether there has been any dissemination effort, and if so, then where, at what geographical units, and we want to document that process, not only the spatial variation, but also the temporal variation of that dissemination, focusing on when is that dissemination happen, when was the initial dissemination took place, and when was the latest dissemination taking place. And we also want to collect more kind of relevant information on the number of manufacturers, distributors, and also the target beneficiaries, mostly whether the dissemination efforts were targeting a specific crop like maize or groundnut, or a specific business like poultry, for example, poultry business or traders, or simply consumers, or sometimes they also focus more on millers and brewery factors. So we want to document both the spatial and temporal dimension of this dissemination, that variation, and also like the variation in the targets. In the second stage, then we want to also get more data on the Aflasafe production, distribution, and usage across all those locations, especially for countries where dissemination has already taken place. Here we want to get more information on the nature of manufacturers. Who are the manufacturers? Are these public or private or public-private companies? If there is any manufacturer, what is the installed manufacturing capacity? And what is the sales of sales per year from the production of Aflasafe? And also want to get more information, if possible, or amount of Aflasafe disseminated, in the price at which the Aflasafe was disseminated, the number of customers reached. It could be farmers, could be traders, or it could be NGOs or projects. And if there is any incentive and rollout mechanism embedded in that dissemination effort, we also want to get that information. For example, whether the dissemination was done through a subsidy or bundled with other inputs, or for free, or by projects subsidized at a different rate. We want to capture that all variation across countries. And for some countries, if possible, then we also want to get a baseline data at the farm or a household or market level, at whatever level the data exists. For example, in Nigeria, from previous project activities, especially by agri results, we know there is some kind of baseline data at the farm level, and we want to document that for the purpose of future impact evaluation. Then why are we collecting this data, and what relevance will this data have for designing impact assessment? So we have some few ideas on how to use this data and why this data could be relevant for impact assessment. But I would be very happy here to get your feedbacks. The first idea we have is right now we are a bit agnostic in a way, because without having good data evidence, we don't even know whether we have to actually conduct ex post evaluation of the impacts, or design a forward-looking kind of impact evaluation to actually measure the effectiveness of scaling options or commercialization options. So this, we want to determine this based on the level of data we have in the type of dissemination efforts we get from the data. We also think that this would also be very helpful for our identification strategy later on, especially for ex post evaluation, because then now if we document those dissemination effects across space and time, then we can actually exploit this variation across space and time in Aflasafe dissemination to estimate impacts for outcomes of interest at the household or farm level. This could also be helpful, we think, that to unpack mechanisms, for example, because if we know the different rolling out mechanisms, for example, with subsidy, without subsidy, then we can actually also exploit this variation in rolling out mechanisms to understand the causal mechanisms, some mediating factors in a way, that contribute to the overall effect of the dissemination. And lastly, the information we think will be also very informative about the key primary and secondary outcomes that should or could be included in our impact evaluation. To give you a sense of what we think the data can be used, we have like 3 scenarios. the first scenario we envisage is actually maybe from this data we could get some kind of sufficient variation within or between countries, mostly within countries, and along with baseline data in terms of the scale of dissemination. In that case, then maybe we could actually think of designing an impact study at a micro level, looking into outcomes at a farm level, production-related outcomes, income-related outcomes, food nutrition level related outcomes, or downside risk due to the better quality, and also to look into both temporal and spatial arbitrage in prices across locations. And we could then use this variation across time and space, or the staggered nature of this dissemination across locations within a given country to identify those effects or to measure impacts. The second case that we envisage is maybe we wouldn't have any sufficient variation within countries, but maybe there could be some variation between countries. So in this case, then we wouldn't be able to actually estimate effects at a household or farm level, but then maybe we could think about an impact assessment focusing on outcomes of interest at national level, mostly macro level outcomes such as total production, trade volume, enterprise development, or if we anticipate any kind of general equilibrium effects, maybe look into the price effects. Then we could then use the temporal variation between countries in a difference in difference or synthetic control estimation strategy to estimate those effects. The third case that we anticipate is maybe that from this data we learn that either there is no variation between or across countries, or uptake is low or at early stage of dissemination. In this case, maybe then we could think of designing a forward-looking impact evaluation strategy, not necessarily an ex-post evaluation strategy. So under this scenario, maybe then we could focus on interventions to alleviate those key supply and demand side constraints for adoption, focusing on maybe commercialization mechanisms, regulatory variation in regulatory environment, such as quality control, signaling, and so on and so forth. And also look more into the cost effectiveness of rolling out mechanisms or commercialization mechanisms. This could be done, for example, using an experimental study, using, for example, it could be an RST at a country level or a specific region in a country. Just to give you some of the lessons that I draw from the process of developing this proposal, I personally learned, kind of appreciated the teamwork and the collaboration and also the discussion we had and I had with SPIA and with IITA socioeconomics colleagues, Aflasafe team, and other and for IA4NH and other colleagues in the CGIAR. So through this discussion, for example, our plans evolved from a narrow scope of evaluating impacts at the micro level, ex-post impacts, to maybe more collaborative and research design with the broader scope. The other important issue that I took lesson of is the importance of understanding innovation-specific properties per se, and then the importance of knowing whom to approach, speaking to the right people, to even think about the causal mechanisms, the kind of impact evaluation we could anticipate. For example, for Aflasafe, if we don't know how it is used and how it actually impacts food production, where, at what level it is applied. Is it a pre-harvest, post-harvest, or at crop level, or at market level? At what scale and at what time? Then we wouldn't be able to actually develop a clean identification strategy, first of all, because we wouldn't be able to cleanly capture all those causal mechanisms and causal chains along the whole value chain. And also, we wouldn't be able to even define some of the key outcomes through which the technology is expected to affect or to lead to impacts. And finally, I believe it's also a learning process because my ideas have evolved over time and for good, I must say, and this is a good option. In terms of design, I believe through this process, it is, you see, like when I started this project, my ideas were to look into assessment at the farm or household level. You know, but sometimes maybe the data, the quality of the data or the type of the data we have at hand might not be good enough or adequate to evaluate impacts at that level. But then we should be able to look into other scales, like other relevant scales beyond farm and household level effects. For example, we could then use an existing data to evaluate effects at the macro level, focusing on macro level outcomes. So this is something that I kind of learned from the process of developing this proposal, and also the importance of defining really outcomes, because for some technologies, complex technologies like Aflasafe, defining simple outcomes is not that easy actually, because the benefits and the costs actually vary over time on space, and it's difficult to quantify those impacts in a clean way. For example, when I first started this proposal, what I had in mind was to look into the cost effectiveness of alternative interventions. But then over time, then I learned that, okay, then what is the relevant benefit to capture, to measure actually, to then quantify the cost effectiveness of a given evaluation strategy, because the benefits actually persist over time and accrued over time, and we can't use actually estimated impacts at a certain cross-sectional point to then determine the cost effectiveness of possible commercialization strategy, for example. So this is something that I learned over time, in the importance of then collaborating, to getting feedback and discussing with others to really articulate those mechanisms. The second point is the importance of really understanding the complexity of the causal chains. Sometimes these causal chains could be long, and also several mechanisms being involved along the causal chain. For example, you see, sometimes we are used to evaluating the effect of a few technologies with clear delineation, like improved varieties. This is much easier to evaluate. But for a complex technology like Aflatoxin, where the causes of incidence are both several and many and long, including both biotic and abiotic factors, then it is very actually difficult to estimate impacts unless we understood all those causal chains and mechanisms. And this, for example, has implications for external validity issues, because then because of the stochastic nature of those abiotic factors, for example, climate, then the effects or the incidence of aflatoxin actually depends on the kind of rainfall, the kind of humidity, and those are stochastic events. And impacts are also stochastic. So we need to take into account the stochastic nature of the factors along those causal chains, actually, to cleanly really estimate impacts for complex technologies like Aflasafe. In terms of evaluation, after going through all this process, maybe then we could think of a more kind of collaborative kind of work where we first map the incidence levels across locations that are currently susceptible for aflatoxin and that are likely to be susceptible in the future due to both biotic and abiotic factors at a larger scale. And if we know this difference variation in susceptibility or incidence levels, first of all, then we can use that discontinuity or variation in incidence across space as an identification strategy. And second, then we could also use some other methodologies like geospatial tools or machine learning tools to predict those incidence levels, and then use those information to actually estimate the overall returns for investment in Aflasafe dissemination, because some of the impacts could actually be obtained from previous or ongoing high quality impact assessment studies on health effects, on nutrition effects, and so on and so forth. So we could actually use those and parameterize, especially explicit, for example, simulation model to actually then determine the impacts, aggregate impacts as a whole. Of course, then wouldn't be able to capture heterogeneity in a way, but then we could be able to actually provide an estimate of the costs and also the returns to investment for such kind of technology where the relevant scale of assessment is not always household or farm, because market level and macro level outcomes are also important. Finally, for discussion and feedback, I would be very happy to hear your thoughts on some of the missing or important indicators that are currently missing in my presentation that could still be incorporated, because we are still at the early stage of proposal development. We have not yet started collecting the data. We hope we will very soon. And also in terms of future strategies, in terms of monitoring and tracking this new and innovative innovations that the CGIAR with potential for future impact evaluation. Is this something that we want to do piece by piece, like Aflasafe, for example, or is something that we should think about at system level in developing a tracking and monitoring system of all those pipeline innovations in a systematic manner, in a way, because we could get economies of scale in terms of cost saving, and also sometimes then we could also involve many CGIAR centers together in a more harmonized manner. So maybe SPIA could think about that. And also in terms of impact evaluation, I see that innovations like Aflasafe and other food safety interventions, or biofortification products, and so on and so forth, maybe we should think about impact assessment focusing on a food system level or a value chain level. And maybe this is something that we should think about within the system transformation, and then how we could actually then use a value chain approach to estimate impacts, not because because of the long causal chains and mechanisms, it's always very hard to unpack all those components cleanly, and maybe we should think about evaluation at food system level. I think I'm done.
Yeah, give some time for people to react and provide some comments. I think Guy Haro has already asked a question here. It's about that you mentioned the possibility of understanding rollout strategies, which is an important question that donors are interested in rigorous evaluation of effectiveness of intervention strategies. Would you be able to say something about this?
Yeah. Okay. Should I respond to the question directly,
yes.
or? Okay.
Yes, please respond while others can raise their comments or suggestions.
Okay. Yeah. So from what I know, from the experience I have in Nigeria, for example, they use a pay for results mechanism to a pull mechanism to roll out Aflasafe. This mechanism used particularly aggregators as incentivizers, like they are incentivizing actually the aggregators per se, not really the farmers, because then the aggregators then could actually reach out to several farmers within their reach. And there is an impact evaluation of that strategy in the final evaluation of the agri-results project. And also, I think there has been some other studies in Kenya, for example, with Vivian and Eric Polte on bundling Aflasafe with weather index insurance. These are kind of like and also maybe an ongoing work, maybe in Senegal with Bernard. Other than that, there aren't really many, much study to inform us on the cost effectiveness of scaling strategy per se, and this could be something that could be then explored from this data, the data that would be collected as part of this effort.
Good. Any other input, comment to Tesfa? Tahir is commenting about the Aflasafe plus the PIC stack as another bundle to write. Karen.
Yeah, thanks, Tesfa, and thanks to the whole team indeed. My question is one, I guess it's first an observation and then a question. So the observation is, I think there is a risk of being a bit too ambitious here and trying to do too many different things at once, while in particular, kind of towards the end of your presentation, and so the kind of going a step at a time, I guess, is going to be useful. I think the logic of thinking that this step can feed into different strategies is useful. I think. And having an open-minded about that, but then in this step, and so this is the question then, it seems to me that one of the, or two of the key things to figure out, thinking about how to use the type of data that would come out of the exercise potentially for designing an impact assessment around, is the same question, which is of catchment areas, although it's a bit funny to talk about catchment areas for Aflasafe, I guess, but it's the same principle. So how have you thought through, kind of, let's say you can track where dissemination happened, through which providers or in which markets, etc. How will that, is there something like a catchment area? Do you expect to be able to define a population linked to a certain market or a certain provider, and what can be done to potentially validate that? And the related question is one about power. So, which you hinted at, right? So I guess the concern, as I understand it, with Aflasafe is low take-up, and so if the potential population of interest is very large and take-up is low, then basically the power in your first stage will be insufficient to say anything about impact. And so partly that's about low take-up, partly that's about the speed of the what is it going to take? How long will you need to collect this data for this to be at a scale big enough to observe impact? So any thoughts on those?
Okay, first of all, thank you, Karen, for the comments. Maybe I will take a few steps on those 2 points, but if maybe Tahir or other IIT colleagues, if you are there, you could also provide your thoughts. So the first point is it something where we could define a subpopulation or a catchment area that could be mapped into? Yes, definitely. So when we in our data collection efforts, especially focusing on spatial dimension of Aflasafe dissemination, we want to collect data not only at the country level, but in which location in that particular country the dissemination has happened especially we want to capture that information as much as possible at the lowest administration or geographical units. Could be, for example, a village. So we'll have a country, a state, or a village, or it could be like a country or an ecological zone, because that's also relevant. And then we could have different locations across. So yeah, in our data collection effort, we want to capture that level of or detailed information, and we believe that if there is then enough variation between those locations within a country, then yeah, that would be helpful for our identification strategy, because that variation could be exploited. That's one of the reasons why we want to get this information at a lower disaggregated geographical unit as much as possible. The second issue is about power. So right now, because we don't know really the scale of dissemination and where those dissemination has happened and the variation in those disseminations, and also the level of uptake perceived in a way. We kind of like we are into in between in a way, because if it's more of like an ex post evaluation, then it would mean that there will be sufficient level of uptake that we could reasonably use for our power calculation. But then what is that sufficient scale? This could be then something that we could discuss with Aflasafe team and others with experience with the scaling of similar technologies. If it's something for that, if our evaluation is mostly on a new study, not really ex post, but like an experimental study testing possible commercialization strategies, then maybe this would be determined based on the suitability of the areas. If we map those susceptible areas in the incidence level where Aflasafe dissemination is likely to have an impact, then maybe we could base our power calculation based on that. But this is something that is at an initial stage, and I think that's something we will have much better thought and clarity as we get more data about the dissemination efforts.
Is there any other comment, suggestion for Tesfa and the team about his design, proposed design? And if not, I mean, you can always reach to Tesfa and the team. I mean, and I'm sure he will appreciate any input that can be provided in the coming days. So if that's the case, probably we can stop here, and I would like to, we have exceeded a little bit the time for the meeting, and I would like to thank everybody to make the time and participate in this one. I'm sure there will be room for many other presentations like this one in the coming seminars, webinars that SPIA is organizing. So thank you everyone for the participation, and especially to Tesfa and Kevin for being the volunteers to start this process and this series of webinar. Thank you very much, everyone.
Thank you. Bye bye.